Rewriting mosquito DNA to end disease spread. Groundbreaking genetic research on mosquitoes. Scientists at Virginia Tech have discovered a new way to identify genetic targets useful for the control of mosquito populations, potentially offering an alternative to insecticides. Their study, published today, July 22, in the scientific journal Communications Biology, focused on the genetic basis of species incompatibility. They crossed A. aegypti, a major global arboviral disease vector, and its sibling species, A. massarensis, from the Indian Ocean. When offspring is crossed back with one parent, about 10% of the progeny becomes intersex and is unable to reproduce. Detailed analysis of genetic expressions and implications. We studied the hybridization of two mosquito species, found that intersex individuals have disrupted sex determination pathways, and identified sex-specific gene expressions, said Igor Sharikov, one of the researchers on the project and professor of entomology and an affiliated faculty with the Fraulein Life Sciences Institute. This study can help identify new sex determination pathway genes that can be used in mosquito control strategies. The researchers looked at 3 areas in their study: the morphological and anatomical level with these intersexes, including external and internal organs, especially reproductive organs, genes involved in the sex determination pathway, specifically 3 genes, 1 master regulator to see whether it's expressed or not, and 2 other genes that create male-specific and female-specific splicing variants. Overall gene expression in those mosquitoes to define what is normal and abnormal in terms of global expression of genes associated with sexual differentiation. Implications for future mosquito control strategies. The study can aid mosquito control by identifying new sex determination pathway genes to create all male populations, thereby eliminating females and reducing vector numbers. Additionally, the identification of sex-specific genes can help develop genetic constructs to manipulate traits in wild populations. Since the intersex is genetically male but expresses female transcripts, it provides a system to identify genes affecting female behavior, which can be useful for future vector control strategies, said Zhengtao Liang, a postdoctoral associate in entomology. Intersexes can serve as a valuable model for the discovery of genetic factors involved in sex determination, sexual differentiation, mating, host-seeking, and blood-biting behaviors in mosquitoes. Finding new sex determination pathway genes and their disruptions in mosquitoes will contribute to the effective control of disease vectors through genetic manipulations based on sex separation.